Hello, everyone, and welcome to today's broadcast, Massively Parallel Genome Engineering, followed by Pooled Growth Selections for Rapid Target Discovery in Microbes, presented by Dr. Laura Klitten, Scientific Liaison at Inscripta. My name is Christy. I'm from Labroots, and I will be your moderator for today's event. Today's educational webcast is brought to you by Labroots and sponsored by Inscripta. For more information on our sponsor, you can click on the sponsor logo on the left side of your screen or visit inscripta.com. Now let's get started. I would like to remind everyone that today's event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many of your questions as we have time for at the end of the presentation. Also, please notice you can share this webinar on your personal social media. Just click on the social sharing tab on the left and let your friends and colleagues know about today's live event. Now, if you have any trouble seeing or hearing the presentation, click on the support tab found at the top right of your presentation window, or just use that Ask a Question box to let us know you're experiencing a problem. I would now like to introduce today's presenter, Dr. Laura Klitten. Dr. Klitten is a scientific liaison with Inscripta's EMEA team. Dr. Klitten is, among other tasks, responsible for correct and timely communication between internal and external partners and acts as the voice of the customer within Inscripta to ensure outstanding support and execution of projects demonstrating the many applications of the Onyx digital genome engineering platform. For a complete biography on Dr. Klitten, please visit the Biography tab at the top of your screen. Welcome, Dr. Klitten. You may now begin your presentation.
Thank you to Labroots for giving me an opportunity to present our beautiful data on massively parallel genome engineering for rapid target discovery in microbes. My name is Laura Klitten, and it's my pleasure to be here today. So the outline of today's talk is that I'll start by giving you a brief introduction to the Onyx chemistry, just so everybody is on the same page regarding the core technology and principles for massive parallel genome engineering. Then I want to show you data on responses to biomass inhibitors in E. coli. I'll show you how we generated more than 25,000 precise and trackable edits across 7 genome-wide libraries, how we then pooled all the cells and performed growth selections in 11 industrially relevant conditions. This pooled growth selection resulted in more than 800,000 data points and response patterns, and I'll show you how we characterize this data for quality and broad response patterns, and how we could create genotype-phenotype maps of responses in inhibitory compounds such as Furfural and vanillin. I'll end my talk by showing you the Onyx platform, how it works, and how easy it is to get started with Onyx in your lab. But before I talk about Inscripta's technology and show you some of the data we've generated, I want you to think about the promise of the bioeconomy. The way we can use biology as inspiration can fundamentally change the way we produce different chemicals, for example, fuels, polymers, flavors, and pharmaceuticals. However, we still have a long way to go for broad-scale implementation since this concept was first introduced around 20 years ago. In order for industry-scale fermentation to work and for the community to increase the number and variety of compounds produced by fermentation, we need high-efficient microbial strains that are more tolerant of either secondary byproducts produced in the fermentation process, or even more importantly, the actual product of interest might be toxic to the cells producing it, for example, vanillin. Vanillin is an important flavor compound originally sourced from the seeds of the Vanillin planifolia plant. However, the industry demand for vanillin greatly exceeds the amount harvested from plants. Currently, aldehydes such as vanillin are produced chemically, but there is an increased demand and promise for more naturally sourced compounds, especially within the food and flavor market. Vanillin can be produced in high-performing and easy-to-engineer microbes such as E. coli, but since the compound is toxic for the cells, and in order to have increased titer and purity of the desired product, we need to modify and optimize the genomes of the strains used for industrial-scale fermentation. During this webinar, I would like you to grab pen and paper and start thinking about how you would design your experiments if you could create 5,000 edits per run instead of 5. If you could create libraries of both genome-wide and rational single gene changes in one go, and if you were free to select exactly the changes you wanted to do. So having an increased scale, increased flexibility, and increased diversity. I hope that by the end of this talk, I've shown you that doing all this is indeed possible, not in the future, but right now. I hope that many of you will send your ideas to me, and we can certainly have a more interactive brainstorm about which projects you would do if you had access to a technology that would enable you to do all these things. Inscripta has developed a massively parallel CRISPR-based and multiplex gene engineering system called the Onyx platform. The technology is powered by Inscripta's own proprietary MAT7, which is a Cas12a family member. It's a programmable endonuclease, and it cuts double-stranded DNA when assembled with the appropriate guide RNA. MAT7 has a distinct and different PAM site than Cas9, and Inscripta's mission is to democratize genome engineering in order for the community to be able to fulfill the promises of the bioeconomy. We've thus made MAT7 free to use for any commercial and non-commercial R&D work. You can download the sequence from our website and start expressing it in your systems today. But please stay for now and see the beautiful data I'll present to you in this webinar, and do not hesitate to write me directly or contact us via our website if you have any questions related to MAT7 or to Onyx. The foundation and core technology was invented by one of our co-founders, Andrew Garst, who has for many years been interested in how to scale up and generate libraries for faster and cheaper genome engineering. This foundational work was published online in Nature Biotechnology in December 2016. It shows how guide RNA and donor arm is covalently linked in order to make sure that the correct guide and donor always are transformed into the same cell. This principle makes it possible to multiplex guide and donor pairs and thus increase precise editing by a homology-directed repair mechanism. As you can see, many others have come up with similar ideas from multiplex library generation in approximately 1.5 years later in the spring of 2018, confirming the tremendous impact multiplex CRISPR editing libraries have on the community. So the core and fundamental, simple yet very elegant concept is the ability to covalently link guide RNA with donor DNA and a barcode for tracking. We are thus moving away from a well-based partition to the single-cell partition. So normally, you would add the guide RNA and the donor template as two separate molecules. And in order to make sure that they are both transformed into the same cell, you would need to have vials or wells as the partition. What Inscripta is doing is having each individual guide RNA covalently linked to the appropriate donor DNA and a barcode for tracking. This concept makes it possible to multiplex all the editing plasmids, and by the unique microfluidics in the Onyx platform, make sure that each guide RNA, donor, and barcode will go into each individual cell. I want to stress here that the barcode stays on the plasmid, and it's only the specific edit which is being incorporated into the genome. This fundamental principle of covalently linked guide RNA, donor, and barcode, together with the microfluidics in the Onyx, gives new possibilities for running trackable libraries in bulk and overcome the need for large 96-well equipment style setup. We are thus increasing the edits that we can perform in one experiment from 96 to thousands of edits at the same time. The reason why we're doing all this is because we acknowledge that there is a gap in what synthetic biologists want to investigate and what is currently technically possible to do. So currently genome-wide methods exist, as you can see on the x-axis here. You can do classical mutagenesis targeting many locations in the genome. However, this is a non-controlled and non-trackable method. It's also possible to generate genome-wide knockout libraries, but here you're limited by the type of mutations you can do. Basically, these libraries only contain one edit type, and that's a deletion. The variety of edit types are depicted on the y-axis. You can, of course, also have many different edit types, for example, in saturation mutagenesis studies of one protein or a few in a pathway. What Inscripta offers is the possibility to reach that upper right quadrant and perform both high variety of edit types and at a genome-wide scale. And that is indeed what Onyx can do. The Onyx platform is a full end-to-end CRISPR-based genome engineering solution, which consists of software, chemistry, unique microfluidics modules for the single-cell work, and a benchtop instrument that would do all your wet lab editing for you. The output from the Onyx instrument is a vial that contains a heterogeneous population of your edited cells, but I'll come back to that in the end of my talk. So here, I'll just show you some real-life examples of different edit types we've generated with Onyx. We can perform edits in both coding and non-coding regions, as well as high variety of edit types. We did a 60 base pair insertion of promoters and ribosomal binding sites in E. coli, and we also did in this study. We performed 38 base pair substitution in the terminator region of yeast and created multiple knockout libraries by introducing triple stop codon at either position 5 or 15 in each gene, and thus creating truncated proteins. And finally, we used Onyx to quickly and really easily generate libraries of saturation mutagenesis for investigation of single proteins or pathways. And the beauty of this technology is that you can mix and match the different edit types into the libraries. So in relation to what you can do with your edited cell population after editing on the Onyx, it's basically possible to move in two directions for phenotyping. You can pool all the cells and grow them up in the presence of, for example, toxic compounds, as we did in this current study. Or you can plate out the cells, grow up individual colonies, and measure the compound you want an increased titer of. It's also possible to basically split the cell population in two parts and do both. So you find both mutations which are giving higher titer as well as better tolerance against inhibitory compounds. Now I want you to show the experiment we did and the beautiful data we generated in this study, where we looked for mutations that would be beneficial and give resistance to biomass inhibitors. In order to transition from petrochemicals to a bio-based economy, we need cost-effective feedstocks at scale. Unfortunately, abundant raw feedstocks such as lignocellulose pose multiple issues. They generally require extensive pretreatment to release fermentable carbon resources, and multiple toxic compounds are released along with the fermentable carbon sources. So many decades of excellent work has resulted in the discovery of resistance pathways and mechanisms to overcome toxic compound inhibition. However, for increased performance and production of new compounds and shifting process requirements, there is a need for rapid optimization at scale. So here we show that decades of work can be recapitulated in a few months with the Onyx platform. And we also showed that even for very well-studied inhibitory compounds, there are huge numbers of additional genomic targets that could be exploited to increase strain tolerance. So we designed genome-wide editing libraries and edited the cells with our Onyx chemistry. So what we did was to design two times knockout libraries by substitution of triple stop codon at position 5 and 15 of all the protein coding genes in the E. coli genome. We then also wanted to see changes based on difference in expression, and we designed 5 different libraries where we took the constitutive promoters of varying strengths from the IGEM database and inserted each promoter in front of every gene in the genome. So what you can see here in the circus plot is actually all the editing designs that we generated in these 7 libraries, from knockout in red to the weakest, from the weakest and to the strongest promoter. So I'm just showing you how we basically make 7 different designs for each gene in the E. coli genome and giving a total of more than 25,000 edits. After the cells were edited with Onyx, we took that heterogeneous cell population and grew it up under selective pressure. So basically, we pooled all our edited cell populations from a 5' promoter of varying strength and the 2 knockout libraries. We then aliquoted the cells into shaking flasks with different known toxic compounds: Furfural, vanillin, hydroxymethylfurfural, and syringic acid, as well as a media control. We also included a gradient of sodium chloride, as osmotic stress is also known to arrest growth. And we added a pH gradient to see how the cell population behaves in acidic as well as alkaline conditions. So as all were run in triplicates, we ran a total of 36 shaking flasks for 48 hours. And after the 48 hours of growth, we harvested the cells, extracted DNA, and created NGS libraries for sequencing. So we know that it can be costly to run NGS in order to track the genomic edits. So Inscripta has developed a plasmid barcode amplicon NGS library prep kit in order to make a quick and reliable measurement of how the cell population behaves. I want to make it clear that the barcode stays on the plasmid after editing. So once you've found your interesting hits, we recommend to confirm these with whole genome sequencing or with sequencing of amplicons from that particular genomic location. In our data, we see a very good correlation between barcode hits and genomic hits, so increased barcode counts will correspond to more cells with that individual edit and thus a growth advantage for that particular clone. Fewer counts will represent reduced number of cells from that clone based on, for example, fitness effects. And we can quickly tell which mutations are beneficial and which are detrimental in a given condition. For sequencing, we typically pool 5 NGS libraries on each MiSeq run, thus sequencing 7 runs on the MiSeq with 1 × 75 base pair setting for this study. So here you can see how the population behaves in the control media. It seems like the stronger promoters gives a bit of an advantage in general, as the normalized counts are increasing with increased promoter strength. So how do you go about and analyze 25,000 edits? First of all, we want to assess the quality and biological reproducibility of the data, and then we determine a method to look for differences in the data among conditions. We look at genome scale trends of enrichment and depletion, and finally, functionally assess the enriched depleted genes to confirm existing knowledge and reveal new findings. We first map the barcode sequencing data back to the genomic edits that we designed, as you see here in this table. In each row, we have the independent promoter or knockout, and in the columns, we have the biological replicates and count data from some of the treatment conditions. This gave us in total more than 800,000 data points. We first then check the reproducibility of the data and see a very high general reproducibility between the biological replicates. We then compare across treatments, and we also see that different toxic compounds are changing the cell population in different ways. For example, it immediately becomes clear that Furfural and vanillin do not have the same effect on the population, as the data does not correlate very well between those treatments. If we just look at the data and how the variance is observed, we see that the biological replicates cluster together and that the cell populations separate based on the stress conditions. We see that the cell population in increased salt concentration alters along the first component, as well as separation on the acidity of the media when looking at the pH gradient. It's clear that the cell population grown in Furfural is very different from the other treatment conditions, including the hydroxymethyl Furfural, as the Furfural population strongly separates on the second component. We can then take a genome-wide view of how the toxic compounds are altering the cell population. Remember that our starting material was the same bulk of cells we then aliquoted into the shaking flasks. So we generally see a difference in the response based on vanillin or Furfural. We typically see both conditions that adding a very weak promoter in front of all genes does not really change the overall population compared to the control media. However, with stronger promoters, we start to see clear changes depending on the mutations and those being more clear. So, and when we look at the knockout libraries, I'll just show them here, we do see clear changes depending on the toxic compounds. It seems that deleting these genes and then adding a vanillin does not really help on the phenotype, whereas in the Furfural experiment, we do see some interesting hits emerging. Another factor for those specifically interested, we also see specific patterns around the origin of replication that can be investigated further. The mechanism of Furfural toxicity is schematically represented here. It's known that Furfural primarily is based on starvation via the NADPH depletion. NADPH-specific oxidoreductases reduce Furfural to the less toxic Furfural alcohol. These NADPH-dependent processes deplete cellular NADPH, resulting in reduced cellular growth. It's known that knockout of the gene YQHD, encoding an alcohol dehydrogenase, which is an NADPH-specific oxidoreductase, rescues cellular growth as a knockout forces Furfural detoxification to occur with NADH-specific oxidoreductases. Furthermore, overexpression of the PNTA gene encoding the protein NADP transhydrogenase subunit alpha helps to alleviate NADPH starvation and will give a growth advantage to these clones in the population. So when zooming in on one specific gene, we can look at how each of the edits we did changes Furfural response. If we look at lactaldehyde reductase encoded by the gene PNTA, we see that adding a constitutive medium-strength promoter will enrich for this clone in the population during Furfural treatment. If we compare the sequencing counts in the control situation with the Furfural treatment, we can calculate the slope and thus have a measurement which will represent a biological response. This is indeed what we see here. The sequencing counts from the 3 replicates in the control media is shown on the lower right corner, whereas the number of cells harboring this mutation are enriched in the Furfural condition as seen on the left. The normalized barcode count is shown on the y-axis. If we then look at all the edits we did on PNTA, we see that the 2 knockouts behave in a similar way with reduced counts in the Furfural condition compared to control. However, looking at the promoters, it seems there is an optimum with the medium-strength promoter and that it does not help to add an even stronger promoter in front of PNTA. Thus, it will give better data if you actually check all types of mutations instead of just assuming that a stronger promoter will be more beneficial for the cells. When we then look at the biological response as log4 changes for all genes in the 7 different conditions, the 2 knockouts and the promoter insertions of increased strength, we see that we cannot, based on this, say that knockouts are always deleterious for the clones, as it for many genes seems to give a growth advantage if knocking out specific genes. We've highlighted PNTA pattern from before in this plot, so you can see whether the cells were depleted or enriched in the population after Furfural treatment when compared to the control condition. We can now cluster the different genes on how they behave in the 7 different mutations. For this, we use UMAP clustering to see how each gene behaves in the knockout and promoter ladder libraries. At the PCA plot, we looked at the variance in each cell population based on treatment, and here we look for genes that behave similarly on the Furfural treatment. So here we just show data from the Furfural treatment and see that genes cluster in overall 4 ways. We observe other genes that behave as PNTA, where the knockout of that gene is a drawback for the cell, whereas increased promoter strength is giving a growth advantage for that cell. In this cluster, we find PNTA and FUCO, confirming what's seen in the literature. The gene ontology terms showing up for this cluster of genes are acetyltransferase activity, general transcription, and cell cycle functions. In the other end of the spectrum, we also see the knockout of specific genes seems to bring a growth advantage to the cells when under Furfural toxic stress. And adding a promoter in front of those genes do not do any good for the cell survival in the population. Sometimes biology makes sense, and sometimes you get surprised about which gene behave like this. So in this cluster of genes, we find functions of carbohydrate metabolic processes and NADPH dehydrogenase activity. In the 2 remaining clusters, we see genes that are either uniformly giving a growth advantage to the cells, no matter what you do, or uniformly have a reduction in growth. So basically, if you are looking to figure out which mutations to introduce into a strain to make it more tolerant towards a toxic compound, this type of data will guide you. And it's even possible to combine the different types of edits into the same cells, thus increasing resistance at a whole new level. But I'll come back to that. If we look at all the edits we generated to find significant changes over the control condition, we find more than 3,000 mutations which are changing the way cells respond to Furfural based on growth patterns. We, for example, find that 2 of the high-strength promoters in front of PNTA has a positive effect when cells are grown in Furfural, as well as knockout of YQHD also gives a growth advantage. So in all, we confirm many previously reported mutations in our study, as well as finding new hits for increased resistance towards Furfural toxicity. If we compare to what is already known in the literature, our study confirms 7 of 8 experimentally validated genes, as well as 253 of 373 predicted mutations from Onyx experiments. We thus, in this study, find almost 2,500 new mutations that have a growth effect upon Furfural toxicity. We can select the most prominent hits, which were enriched and thus showed the highest fold changes between control and Furfural treatment. Based on this data, we can now select our most promising strain, revalidate it, and use this as a starting strain for next round of diversity generation, for example, by combining the winners or simply do a second round of genome-wide agnostic mutations so we can perform combinatorial optimization. This way, we can quickly jump the fitness ladder and dramatically increase tolerance towards toxic compounds or increase the titer of desired product if that's what you want to optimize. It's of course also possible to do both, and we've actually done so in a project where we optimized lysine production and looked for increased tolerance against thiolysine, which is a toxic biometabolite in the lysine amino acid biosynthesis pathway. So today I showed you data where we designed and generated more than 25,000 precise and trackable edits across 7 genome scale libraries in a really easy and intuitive way. We performed growth selection of these libraries in 11 industrially relevant conditions. We characterized more than 800,000 data points for quality and broad response patterns. We performed genotype-phenotype maps of response to inhibitory compounds, Furfural and vanillin. And this study, we confirmed existing literature as well as discovered hundreds of novel targets for strain improvement. I want to now show you how easy it is to create high-throughput, massively parallel genome engineering libraries with the Onyx platform in your own lab. Inscripta's Onyx platform supports all areas of the design, generate, test, and learn cycle. Our online software, called Inscripta Designer, will design guide RNA and donor DNA pairs and assign a unique barcode. You simply inform the software what edits you want and where you want them. We've built an intuitive graphical user interface, and if you are more bioinformatics savvy, you can also upload a CSV file with all the edits you want. Inscripta Designer will provide a color code for each design based on more than 1,000 different parameters in order to guide you in relation to how good each particular design is. You can then select the designs you want and test, and we will manufacture the custom reagents containing your specific guide RNA, donor DNA, and barcode in complete kits to drive genome engineering. You will then receive the custom reagents and the microfluidics consumables, which fit into the Onyx benchtop instrument that you have in your lab. The footprint of the instrument is very small. The instrument will take care of all wet lab parts of the editing experiment. The on-instrument editing takes 2 to 3 days, and the result is a heterogeneous cell population of minimum 1 × 10⁷ cells. We've developed NGS assays for both immediate QC of the editing experiment and for downstream analysis of the population after phenotyping. Once phenotyped, it's possible to track back any barcode sequencing to the genome level and upload this data into our online Inscripta Resolver software, which will help you analyze experimental results and thus guide you for the next round of the DGTL cycle. I want to pause here and just explain to you how Inscripta only calls edits if they are complete and intended. You see here on the left panel an example of what we mean by complete and intended. In this experiment, we wanted to change the codon for tyrosine, TAC, to threonine, ACT. So even though we still encode a threonine with the ACA, it's complete, but it's not intended, and therefore not included in the metric. You can also see that we create auxiliary edits in the PAM and spacer regions to prevent recutting of the MAD7 endonuclease. Here you see on the circus plot, it's just an example of one single guide RNA targeting chromosome 1 of a yeast genome, and with the height of the bar indicating the editing rate, and thus the fraction of cells in the population with a complete and intended edit on that specific position. When comparing different metrics with literature, it's important to think about what is meant by that metric. Typically, mechanisms of both non-homologous end joining and homology-directed repair are reported as successful editing. But when you move from single edits to massive parallel libraries of guide RNAs, we need to be able to exactly trust that the edit we designed is also what we see in the final population. Therefore, we only count edits repaired by homology-directed repair at the specified genome position. So each guide will, dependent on position and edit type, behave a little differently. So an Inscripta reported edit efficiency is the number of cells in the cell population which has a complete and intended edit, whereas the designs observed is showing the number of designs we find in the population. I'll just give you an example of what these metrics mean. If you have 50% of your cells in the library with complete and intended edit and a 99 design represented in a library of 10,000 designs, we can, per 1 million output cells, expect to find 500,000 cells in the population with a complete and intended edit. Of those 500,000 cells, we will find 9,900 editing designs at least once. So here you see the specs of the Onyx platform. The technology has been developed for E. coli in yeast. It's optimized for Inscripta standard strains, and we can also use custom strains on the instrument. We perform 1 edit per cell, and those can be insertions, deletions, and substitutions, and a combination of those in the libraries. Each library can generate from 100 to 10,000 edits for E. coli and up to 6,000 edits for yeast. So if you want more edits, as we did in the data I just showed you, it's possible to pool cells from different libraries and phenotype everything together. The edit efficiency is 30%, but varies from 15 to 75, depending on the specific library produced. DNA-based editing libraries and other reagent manufacturing is 3 to 4 weeks, and the on-instrument time is only 2 to 3 days. So I want to end here by thanking all my brilliant colleagues in the applications development and product development teams who designed the experiment, did the lab work, and all the data analysis. And I just want to thank you for attention, and I'll be happy to take any questions.
Thank you, Dr. Klitten, for your informative presentation. We will now start the live Q&A portion of our webinar. Now to our audience. If you have any questions you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. We have lots of questions already coming in. Dr. Klitten, let's start with this first one. When tracking genome edits using the plasmid barcode, how confident are you that the edit has been made?
So thanks, Christy. That's a great question. We are quite certain that the edits are made, but of course you cannot be 100% sure because, as I said in the presentation, that the barcode stays on the plasmid. We don't want the barcodes on the genome. So we recommend to actually reconfirm that you have that genomic edit, especially if you have some interesting strains that you are looking for. And you can do that with whole genome sequencing. You can also do amplicon sequencing of that particular region if it's just a few edits. But you can actually, we also developed a pooled whole genome sequencing assay for to actually do the whole pool. So it makes it really easy to see the genomic edits. Yeah.
Very good, thank you. Our next question, would it be possible to explain in a bit more detail how your editing protocol allow for the generation of both a high variety of edits and across the genome?
Yeah, so this is basically very kind of, this question is very kind of core of the technology, because that's actually what Onyx is about. It's to have that ability to do both. And that's why we've developed this holistic way of like end-to-end solution, because you need very good guide RNA design. That's very important to really get that. And you need the homology arm. You need that to happen very efficiently. But you also need to make sure that you're not, introducing something that you don't want. So we worked a long time actually to develop this technology. So that's why we have the instrument that is running in the lab, and we have the microfluidics components to make sure that we do that we are able to design these thousands of edits in one library, and that we can do both the substitutions and the insertions and the deletions, and you can basically mix that up into every library. And because this is working in single cells, that's the reason why you can actually do this.
Thank you, Dr. Klitten. Our next question, are cells alive post-experiment? Can you isolate them again for large-scale fermentation? And how is the DNA extracted for library prep for Illumina-Seq?
Yeah, so yes, they are alive because we are working with living cells, and that's what this is all about. And yes, you can also isolate them afterwards. So you can grow them up as clonal isolates, to actually be able to get that specific clone and grow that up for any like other experiments. For example, if you want to do like the next round of diversity generation, you can use that strain. And it's also possible to, like, if you want to confirm your edits, you can actually also prepare, like, create a small library. If you, for example, have maybe 100 like clones that you found, wow, these were really good, you can redo this on the Onyx and then grow up those cells, because then you have more cells in your population which harbors those kind of 100 edits, instead of if you have a large library, because you need the high coverage, you will of course have fewer cells which has that particular edit in it. But yes, it's possible. They are alive and you can isolate them. And the kit we did for this particular study was just a standard QIAGEN DNA isolation kit. But we've also made an application note with lots of more details around this particular data that I showed today. And please let me know if you want a copy of the application note, and I will be very happy to send it to you. And there are lots of details about how we, the methods and so on.
Dr. Klitten, how will your method support hit validation if I want to reconstruct edits which have shown to be enriched in pool cultivation experiments?
Yeah, so I talked about that a little bit, but I think it's important that you can do like validate if you, for example, have found a number like 100 hits or so, you can actually create a small library on the Onyx in order to reconfirm this and just do sequencing of those fewer different designs. So it's also possible with the MAD7 in your lab to do that like single, like for any like single work. But since we are working, so Onyx is about high throughput and this is about scale up. So we would recommend to actually pick the best 100 and then do a small library for validation and to have your cells with that particular edit.
Thank you. Do you plan to enable insertions of large DNA molecules, such as heterologous genes and pathways on the Onyx platform?
Yeah, so this is a question that we get a lot, and of course we want to be able to support larger inserts and larger inserts in the future. Right now, the technology allows us to do between 1 and 60 base pair. And the reason why we haven't just said, well, you can do thousands, is because we want to make sure that this is actually efficient for every design that you're making. So it has to be efficient for the whole genome, not just one particular region, but it has to be efficient all over the genome. It has to also, the coverage of the different designs they have to be well represented in the library. So when you get your cells, that the designs that you made is actually also present in the cell population. So that's the reason why we're not offering these large inserts right at this time point, but this is something that we're definitely looking at, and this is something that is on our roadmap to come.
Thank you, Dr. Klitten. Now we do have time for a few more questions, but I do want to let our audience know any questions we're unable to answer live today. And additionally, those that come in during the on-demand period, they will be addressed by our speaker via the contact information you provided at the time of registration. Okay, let's keep going. Our next question, genome-wide libraries seem like a powerful approach to diversity generation. Do you plan to offer pre-made knockout libraries?
Yeah, so this is a great question. Right at this point in time, we actually see that the need to be able to mix and match the different edit types. So basically, I guess one of the things we want is people to think about not being limited by the way they want to do libraries. So we're not at this point in time offering any like pre-made libraries, because we want to make sure that people understand that this is actually a matter for you to exactly design, I want these knockouts, and I also want these promoters, or I want these saturation mutagenesis of that particular protein, and you can mix all that into one library. And in order to do that, we need to have this as a custom order, because every customer is different and everybody is looking at different things. But of course, if you are working and you want to kind of have a lot of the same types of libraries, you can certainly contact us and we can figure out a way to like having those kind of pre-made. So that's definitely something we can talk about, but right now it's not like an off-the-shelf offering.
And Dr. Klitten, this question's come in a few times. Can you apply this approach to other organisms and viral genomes?
Yeah, so this is a great question. So right now, Onyx is optimized for E. coli and Saccharomyces cerevisiae, and we want to make sure that the editing efficiency is high, and that the coverage is also high, so the design coverage. So right now, you need to use one of those organisms as your kind of workhorse, if you can call it like that. So if you, for example, have a human gene or a viral gene, or you want to kind of look at anything specific, you can work with that and do heterologous expression in E. coli, for example, or in yeast. So that's the option we have at the moment. We are looking into which other species should be made available on Onyx. But as you can imagine, since this is biology and these are live cells, so every time you move into a new organism, you have to kind of optimize the platform again for that, because it's very important for us that this is a high efficient and very well working technology. So right now, I would recommend to see if you can kind of heterologically express your proteins of interest into an E. coli or yeast in order to actually have benefits of the massive scale that Onyx offers.
Thank you, Dr. Klitten. And we have time for one more question. How do you select for edited cells to avoid amplification on non-edited cells?
Yeah, so the pool will, of course, always have, there will be some cells are edited and some are not. And we do have on the plasmid, we have antibiotic resistance markers to make sure that we are, as long as you are adding your antibiotics, you will still have a preference growth of any cell that harbors the plasmid. But ultimately, we of course want the specific edits to be the growth advantage. But at this point in time, you can use antibiotics and grow up with these resistance markers in order to have higher number of just the overall population. Yeah.
Thank you, Dr. Klitten, for this excellent Q&A session. Do you have any final comments for our audience?
Yeah, so I just want to thank everybody so much for staying today and attending this webinar and listening. I hope that you felt this was time well spent, and that I just want to let you know that we also offer digital seminars with our science team, as well as physical seminars when when it's safe to travel again, then we can also come and visit you and do seminars. So I just want to encourage everybody to write me, or and if you want that, or you can also go to Inscripta's website and fill in the contact information. If you are interested in having any seminar or discussing more about the technology, we are very happy to do so.
Thank you. Now, before we go, I do want to thank our audience for joining us today and for their interesting questions. We did have quite a few questions we were unable to answer live today. So those questions, and additionally those that are submitted during the on-demand period, they will be addressed by Dr. Klitten via the email address you provided at the time of registration. I'd once again like to thank Dr. Laura Klitten for her time today and her important research. We would also like to thank Labroots and our sponsor, Inscripta, for underwriting today's educational webcast. Now, today's webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Have a great day.